Welcome everyone to this 2024 seasonal update webinar for pulses and mung beans, being presented to you by the Australian Fungicide Resistance Extension Network, also known as AFREN. I'm Bridget Penner from Ag Communicators, and in today's session we're pleased to have the leading experts in fungicide resistance, mung beans and pulses here to present this update for you. So first off, we'll hear from Fran Lopez Ruiz, who leads the fungicide resistance group at the Centre for Crop and Disease Management at Curtin Uni. He is the guru on all things fungicide resistance. Then we have Levente, who is a professor of plant pathology at the University of Southern Queensland and the director of the University of Southern Queensland Centre for Crop Health. Sorry, I've got the order wrong. We'll then hear from Josh Fanning, who's the plant pathology research leader at AgVic in Horsham, and then we'll hear from Levy. So for those who are not familiar, AFREN is a GRDC-supported initiative that provides up-to-date resources and information directly to Australian growers, agronomists and advisors. You can find a whole heap of resources on the AFREN website, including fact sheets, workshops, webinars such as this one, podcasts and more. So before we begin, just a brief word on housekeeping. We've got a Q&A function available for today's webinar. So if you have any questions that crop up during the presentations, feel free to type them into the Q&A box and we'll address those at the end of the session. So without any further ado, I will hand over to Fran. Over to you, Fran.
Thank you very much, Bridget, and thank you everybody for joining this webinar today. I'm going to be talking a little bit about fungicide resistance. It's going to be very brief because I don't want to take time from Josh and Levy. I guess that the first question is to understand why do we have a problem with fungicide resistance, and not so much, or not in the field at least, for pulses. We use fungicides because they work at controlling diseases. They help us managing diseases and improving yields. The question here is to use fungicides in a rational way, and this is what AFREN is all about. So fungicide resistance is not selected by fungicides. So resistance is something that already exists in nature. So we use fungicides, and by using fungicides, we select for resistance. In this image, I'm just showing a leaf that contains pathogen populations. Blue ones are those that are sensitive for the fungicides, and the black ones are those that are resistant to fungicides. When we spray a fungicide, what is going to happen is that the majority of those individuals, sensitive individuals, are gonna be controlled. Some of them will survive because remember, we never eradicate, we manage diseases. Their numbers are so high to eliminate them all. Often we spray again. And unfortunately, in many cases, that spray is very similar. If it is not the same formulation, it's from the same group of fungicides. And that second spray, or third spray, or fourth spray, what it's going to do is select those resistant individuals that now have an advantage over the sensitive fungi. So these individuals are going to increase in frequency in the population. And if we keep doing that over and over again, we end up with an outbreak of resistance. We have cases of these outbreaks in Australia a few times already. So, but actually, fungicide resistance is not the main reason why a fungicide doesn't work well in the field and doesn't control the disease. Is one reason, but it's not the main one. There's several reasons that are not related to fungicide resistance that are behind the fungicide not working well. Like for example, the lack of genetic resistance in the crop that we are using. So if our varieties are very susceptible to disease, we cannot expect the fungicide to work perfectly and control beautifully a disease in the crop. So we have to expect, some level of disease. Poor IDM practices, like not managing stubble, not controlling the green breach, not rotating the crop. So all these management strategies, if we don't deploy them properly, so obviously the disease level is going to be higher. So fungicide is gonna struggle at controlling that increased level of disease. Using the wrong product at the wrong time, not having a proper coverage, reducing water volumes, that's also going to impact the performance of the fungicide. And finally, the one that I consider the most important is the conducive season. So if the conditions are conducive to disease, very conducive to disease, we're going to have more disease. More disease means that the fungus is going to have to work harder at controlling it. So on the contrary, if the season is not conducive, so well, we're going to have less disease and less chance to have to use fungicides. This slide here is just showing this very busy slide, sorry, showing a very convoluted summary of what's the situation in terms of fungicide resistance in Australia at the moment. And there's a purpose why the slide is complex, and it's because the situation is quite complex. There are many cases of fungicide resistance, but not all crops are affected. And actually, this is a situation for pulses and mungbeans. So we've got some detections of resistance in the field. Some mutations are being associated with resistance, particularly botrytis gray mold chickpeas, group 1, ascochyta lentils, group 1 as well. Mungbean, as Levo was talking about later, group 3 and group 11. We don't have field reports from the field. We don't have field reports indicating that fungicides are failing in the field. The reason why this is not happening is probably because the frequency of these mutations in the field is still low. But obviously, this is a very important warning for us. Those mutations exist. And if we keep using fungicides without the techniques that we are explaining in these webinars and all the information that is delivered through AFREN, obviously the frequency of these mutations is going to increase, and we will end up having issues in the field. And I believe that this is my last slide, and now I'm just passing the webinar on to Josh. Thank you.
Thanks, Fran, for that. Firstly, I'd just like to acknowledge Curtin University and the Centre for Crop Disease Management for making this happen, along with Ag Communicators and GRDC for the investment in this area and co-investment. So this presentation today is national, and so I thought I'll start by putting up a range of pulse pathologists, and I've talked with a number of these prior to this presentation to see what's happening across Australia. And we have got a range of seasons occurring across Australia, from a very good season in most places in Queensland. Large amounts of chickpeas have gone in as a result of tariffs being lifted in India. And essentially in New South Wales, there's been high rainfall up in the northern areas, or in a lot of the northern areas. We're not seeing huge amounts of disease. Chickpea ascochyta blight and a bit of botrytis is just starting to develop in those northern regions. From talking with the pathologists up there, we've got a little bit of chocolate spot in some of the field experiments that we've got going. Moving into southern New South Wales, where the season's not too bad. They've had good soil moisture to start the season, and essentially good rainfall within the season. Then moving into Victoria, we've got a range of seasons occurring again, even within Victoria. We started off with good soil moisture in most areas in Victoria. Some areas had an early start, but a lot of areas had a very late start. So we've got a range of plant growth stages through the season. Some in the Wimmera certainly haven't reached canopy closure, whereas other areas have definitely surpassed canopy closure. In-season rainfall has been highly variable as well. And in the Wimmera, it's been quite conducive for disease in the last couple of weeks, and especially in high-risk environments, we saw certainly early ascochyta blight and lentils. In South Australia, we've got a bit of a contrasting season to Victoria. They started with no soil moisture or very little soil moisture, and they've been lacking a lot of in-season rainfall. So some areas are certainly struggling, and disease isn't as big a constraint in South Australia from what I'm hearing. WA has a mixed season as well. Northern areas, they're starting to see sclerotinia and lupins and canola. And a lot of the other areas, they had a very late start, but they've got very good seasons in a lot of areas even after that late start, but highly variable across there. So today I'm just going to go through some of the general disease management techniques across most of our crops, or the ones that I know a lot about. And where a lot of the research has been occurring across this group of pathologists and other pathologists in there. So as a brief overview, I'll go through some disease basics, I'll go through some common diseases, integrated disease management, and then fungicide resistance management, because I really don't want pulses to end up similar to that first slide that Fran put up, with a very convoluted mess of basically fungicide resistance over a range of crop by diseases, essentially. Whereas in pulses, we're quite lucky at the moment. A lot of our fungicides are still highly effective. But we want them to remain that way, so we don't want to overuse them by basically reducing that chance of resistance developing. So Fran talked about why we think fungicides aren't working in the field, and I think this slide here brings that back in together, looking at the disease triangle essentially, where you've got to have 3 factors for disease to occur in the field. And you remove one of these factors, then we're not going to see disease. And it informs the risk the whole way through the season. So if we look at the pathogen, we're looking at how that pathogen survives from season to season. Pathogens cause disease, but without that pathogen, we just can't have disease. Essentially, the infection cycles, how is it dispersed and how does it reproduce? And then during the season, we've got our environmental factors. So if the disease has come through, what temperatures, humidities, rainfall have we been seeing? Foggy conditions certainly cause disease. They cause leaf wetness. And then soil moisture. A lot of us forget about soil moisture, but if we've got good soil moisture, that's releasing water back up and making our canopy quite humid in some conditions, which aids in that disease development. And then we've got the host, and that's really our varietal structure, but also varietal resistance, but how does our canopy grow? Some of our crops have very different canopy structures, which then obviously influences the microclimates in that. And then what growth stage is the crop at? So that's what we're targeting usually for our fungicides, but it's also when is the disease likely to come and when is the disease going to cause an issue? So if you don't remember anything else from the presentations, I think this slide's probably the most important. It's starting with our building blocks for disease management, and essentially starting with that host resistance, selecting the most resistant possible variety or the least susceptible variety or crop that you can in that environment. And then, so you're not relying on the other methods during the season. Then we've got our non-chemical options. So that's our disease carryover from season to season. So how are we managing our rotations, our stubble? When are we sowing? Sowing early might mean plants are out of the ground a lot earlier, so they're exposed to the environment and potentially the pathogen for a lot longer. And then in some diseases, it's about managing the green bridge, such as rust, but that's not so much a concern in our pulses. And then we should be relying on fungicides last. I know they're cheap, and especially this year, they can be quite cheap. But essentially, if we rely on them totally at the moment, then you're basically going to end up, like what Fran said earlier, with lots of resistance. And then we're going to be worrying about how we're going to control it when the pathogen does overcome our variety resistance later. And it's about using mixtures if available and staying within label rates. So I know this presentation's about disease, but I think the first part is it really disease when you're looking in a field? The picture on the screen there is of physiological spotting in chickpea, but we really need to know and need to accurately diagnose whether we've got disease and whether we need to manage it. Is it virus? Is it an abiotic stress? Is it water stress? This year we've got a lot of areas that may be struggling water-wise, so if our root structures aren't right, are they getting enough water and are they exhibiting symptoms which are essentially water stress or a lack of nutrition? Or without that water and rainfall, some of our herbicides are having an effect on our plants, causing symptoms as well, and essentially could be misdiagnosed as disease, and we're applying fungicides unnecessarily, which is not only a cost, but it also causes selection pressure on our pathogen, leading to potentially resistance in the future. So soil-borne diseases, I want to touch on them briefly. It's not so much a concern because there's very few fungicides that are available and seed treatments available for soil-borne diseases, but it comes back to that slide before. Are we correctly diagnosing what we're seeing? If we're seeing some of those soil-borne diseases there, have they impacted our root structures, which is then limiting our moisture into the plant? And are they causing other symptoms above ground that can be misdiagnosed as disease? So what are our common pulse diseases? I sort of put them into three main categories across most of our pulses. We've got botrytis, sclerotinia, and ascochyta blight. There are three main diseases. In lupins, we don't have ascochyta blight. We've got brown leaf spot. And then in faba beans, we've got Cercospora, field pea, we've got bacterial blight, and we've got a few other diseases that we don't see so often in lentil that might be affecting as well. So I'm going to go through focus on the three main diseases today in this presentation. So ascochyta blight, some of the pictures of the symptoms are there. It causes yield loss. We're sort of thinking up to that 10, 20, 30% yield loss. If we're seeing stem breakages, we can see, especially in young, vulnerable seedlings, we can see basically seedling death. Does that result in yield loss? Hopefully some of our current investments will be able to tease that out. If we look at the top picture on the left there, it's a beautiful lesion for a pathologist, but essentially you're talking a white necrotic tissue with black pycnidia or black dots in it. And you can see that in those pictures on the bottom, on chickpea, lentil, and on a lentil pod. And then you can see the effects on seed up there on the top right. So it does cause seed staining. It can also cause downgrading of that seed quality at the end of the season across our crop species. Ascochyta blight has a separate ascochyta pathogen for all our crops. So that means we don't need to worry so much if we're rotating between a lentil and a faba bean or a chickpea. Ascochyta doesn't go between those crops. And that'll become more important when we talk about botrytis later. So the life cycle of ascochyta blight, I don't want to go through the full life cycle, but essentially it's really important because that's how it carries over from season to season. And if we look down the bottom there, we can see infected seed is one transmission method into the seedling, and the other one is infected crop residue. So that's how it carries over from season to season. And then if we move up towards the left, you've basically got your pycnidium and then cnidia, and basically from those pycnidia that we see on the plant, those lesions, then we can have rain and wind causing those spores to travel up into the plant, and that's how it spreads very quickly within the season. Now, chickpeas are as our most aggressive, where we've got a lot of yield loss data. So here's some Victorian data showing that basically we can have complete crop failure in some environments. So Cario was a low rainfall zone, but we still saw 96% yield loss in a wetter season up there. But we see a range of yield losses. Something that's quite important there, though, is if you look at the susceptible variety on the right, PBA striker, you're seeing quite significant yield losses there. Whereas if you're looking at or complete crop failure in some seasons, whereas if you look at the moderately susceptible variety, so all we're doing is getting rid of the suckers or reducing the susceptibility of our variety, and we're seeing a dramatic yield increase. Now, yes, they're not acceptable yield losses by any stretch of the imagination, but what we're doing is we're starting off with a much better resistance as a building block, which will then require less fungicides than our susceptible variety. And this is common across our crops. The yield losses certainly aren't what we see in a lot of the other crops, though. In field pea, we did see complete crop failure in 2022, but the best data I've been able to pull from trial results was from some SARDI data from a number of years ago from Jenny Davidson. And we're talking sort of a half a ton yield difference in most seasons of fungicide, a full fungicide regime, which would certainly not be economical versus a no fungicide. And often we can get away with just that essentially early flowering application of fungicide. And we have got better fungicides available to us now. But if we've got that early fungicide at that 8 node stage or early flowering as well at times, essentially we can control that disease. And it's about monitoring those crops, which I'll go into after. So ascochyta blight management is mostly about choosing resistant varieties. We're very lucky in our pulses. We have very good resistance to ascochyta blight across most of them, apart from chickpea. But there's certainly some great varieties coming in chickpea and what I'm seeing in the field and what Christy's telling me and what we're seeing through the program. So it'll be exciting to see those varieties come through in the next few years. But keep in mind the potential value of the crop and potential yield losses. In lentils, we don't seem to see that massive yield loss, and in vetch, we certainly don't know the yield losses, but we don't suspect that we're seeing anywhere of those yield losses. And what's the final stage of that crop? What are you actually going to do with that crop? Planting away from last year's stubble. So I know at this time of the year, we're still starting to think into next year. We're doing planning for next year. So planting away 500 meters from your previous crop, if you're heavily infected with ascochyta, is certainly a good recommendation. And then for field peas, there's Black Spot Manager of Field Peas, which Deep-Herd release every year. It gives you your risk based on sowing and the rainfall that you've had prior to sowing. So I'd certainly look that up if you're growing field peas, and subscribe to that service by Deep-Herd. Fungicides, be mindful of the mode of action, systemic versus protectant. Essentially, we say foliage that's been sprayed, that hasn't been sprayed, is unprotected. Some of the new chemistries show a little bit more of that curative effect, but it's certainly not what we recommend. And is it curative in 24 hours or 48 hours or 36? We don't know, because we haven't done that, because everything we do is based on protection. Chickpeas, it's really important to spray before the rainfall event and keep on top of it. Ascochyta in chickpeas, we know it in 10 days or 7 to 10 days. Essentially, it can go through a full life cycle. It's very highly aggressive and we're lacking that resistance. So potentially you'll need to be spraying fortnightly when leaf wetness is common. Botrytis grey mold's the next one that I was going to talk about. So here we've got some lentil botrytis grey mold. It's grey fluffy mold there in the center pictured. We see white lesions on the leaves without those black pycnidia, and then it can certainly affect the pods in a similar way, and then seed quality as well. It's called chocolate spot in faba beans. Looks slightly different, so we're not seeing those white lesions. We're looking at the chocolate coloured lesions. Certainly affects seed quality as pictured on the right. But how do we tell chocolate spot from other lesions that we see on beans? Beans get a lot of lesions, or what we call lesions or spots. My telltale sign is when you go to the flowers there and you look at those pictures of the flowers, you can see the chocolate spot lesions on the flowers. And the flowers are quite susceptible to chocolate spot. So that's when we know we've got chocolate spot and botrytis. And botrytis is a very aggressive disease, more aggressive than chickpea ascochyta. The other thing about botrytis is it goes, the pathogen goes between crop species. So botrytis cinerea affects all our crop species. And sorry, I've left lupins off there. But botrytis cinerea goes across everything, and botrytis fabi goes across our vetch, faba beans, and lentils. So that's really important if you've got a vetch crop for the southern region that's right next to a crop of faba beans or a crop of lentils, that you might go, oh, I don't care about vetch, the end use, I'm just going to spray it out, but it's showering spores onto your highly profitable crop next door. And that might be the case in Queensland, where you've got chickpeas that you've got botrytis in one paddock that you haven't been able to get on to spray. It's going to be showering spores into potentially other chickpea crops around it, or other crops around it. So it's important to control this disease early to protect neighbouring crops as well. Sowing density and that microclimate is really, really important. This graph here shows a botrytis grey mold score. And basically 40 to 200 plants per square metre. We've got a susceptible variety on the left and a resistant variety on the right. We can see the resistant variety. It doesn't matter what sowing rate you have no disease. It's a resistant variety. It's fantastic. You don't have to worry about disease management in it. Whereas if you look at the variety on the left, the greater number of plants, basically you've got more density of stems, more density of leaves, and therefore you've got a more humid environment there. You've got canopy closure usually much sooner, so the fungicide's harder to get into the canopy, and we're seeing more disease in an untreated fungicide plot. Variety resistance, just to hammer that home further, if we look here at beans, we've got Fiesta on the right, 0.72 tonnes per hectare, whereas PBA Amberley, yes, it's not resistant, it's MRMS, yielded about 4.5 tonne with fungicides in this experiment. But you're talking a massive difference in yield just due to varietal resistance. That's really why we push this. Your varietal resistance is really that sounding block, right? That building block right at the start. Economics of fungicides. We've got a range of fungicides that are available for us. This graph here shows gross margin, and we've got some active ingredients on the left, starting with Veritas, tebuconazole, azoxystrobin. We've got prasimidone, Group 1 fungicide. And then, Group 2 fungicide. Carbendazim also on the right, down the bottom. And then in the middle, we've got aviator, bixafen, prothioconazole. And then in the middle, the nice long one, really good profitability, fludionoxyl, pyroflumetriphen, miravis. But interestingly, carbendazim down the bottom, we mistimed a fungicide application in this experiment and actually had it at canopy closure, whereas our prasimidone was at flowering, and so were all our other fungicides. First fungicide at flowering. If we get on top of this disease early, essentially we can control it, and therefore we can have really good gross margins. So that's the take home there. It's not necessarily about what fungicide you use, it's about the application method. So in botrytis, agronomy is just as important and choosing a resistant variety. But really fungicides are essential for botrytis because we don't see the level of resistance in a lot. And I mentioned in PBA Amberley for 2.75 tonne yield there in that trial untreated. But when we added fungicides, we're up to 4.5 tonnes. There are some varieties, but they're starting to lose that resistance. So we need to be proactive and keep on top of it. We need to think about our fungicide efficacy and fungicide coverage. So we're trying to go before canopy closure, because then essentially we can cover the whole plant with fungicide. And then we're applying fungicide as required after that. But environment is key. So if you're in a lower rainfall zone this year, you're unlikely to see botrytis pop up at all, because you're not reaching canopy closure or you're not seeing those warm, humid conditions. It's similar to ascochyta blight. If you're not getting the rainfall, the best fungicide is dry weather, essentially. Sclerotinia white mold's the last disease I'll cover today. There's just some pictures up there. It can be quite devastating across a paddock scale, as opposed to botrytis grey mold, which I just talked to. That's a grey mold, as it says in the name, whereas this Sclerotinia is a white mold. So you can see the white mycelial growth across the surface on the bottom left there. And then it's carried over from season to season with the sort of black mouse-like droppings on the right. And then there's a little mushroom there. And basically that that's another way of carrying over the disease, or it grows out of the black sclerotes. Essentially, you get that coming out. And it basically showers spores up into the crop later. Sclerotinia white mold is caused by a number of pathogens, but really essentially there's a really broad host range. So all your broadleafs cover it. So lentil, chickpea, field pea, vetch, canola, lupin as well, sorry, and then your weeds. And those sclerotes last up to say 10 to 15 years. People have tried thrashing them through a header. They just break the sclerotes up to smaller and help with the spreading of it. So essentially, you've got to monitor what paddocks have Sclerotinia in it. In terms of fungicide efficacy, we don't have too much data on that at the moment. So we're really looking at how that spreads between season, and really realistically, we're looking at those sclerotia, the black mouse-like droppings between seasons. It can survive in crop residues, but really it's that sclerotia from season to season that last. And then those little mushrooms, the apothecia that come up, are basically our in-season spreading of that disease. So for Sclerotinia, we don't know huge amounts about the fungicides. We are seeing certainly effects of fungicides and a 5 to 10% yield increase due to fungicides. Is that economical? It'll really depend on your situation and how severe the disease is. So we really recommend an integrated disease management approach at this time, which is what we recommend across all our diseases. And so that approach should be starting with sowing clean seed, so it doesn't have those sclerotia at the start, it doesn't have ascochyta infected seed or botrytis. It's thinking about our crop rotations and what pathogens we saw last year. So you're not going to plant a lentil on a faba bean that was covered in botrytis, because that's not going to be good for our rotations. Choosing our resistant varieties, monitoring for our disease, understanding our disease risk, and then optimizing our fungicide strategy. So we're not going to be applying as many fungicides to resistant or moderately resistant varieties as what we are to susceptibles, or we may not need a fungicide at all. We're spraying before rainfall events. and really understanding how many rainfall events are there and what's your seasonal risk. That's really important because that risk underlines how many fungicides you're going to require, and that with your building block of resistant varieties. And then remembering plant growth post-spray is unprotected and sprays are preventative, not curative. And again, back to protecting our seed. We want to make sure our seed's protected at the end of the season. So we might need a potting chlorothalonil to basically prevent seed-borne infection. Varietal resistance, where do we find that data? There's the Pulse Disease Guide in Victoria, the NVT ratings website. I was on the internet the other day looking, and the Pulse Guide from Western Australia is really, really good. A lot of our states have these Pulse Disease Guides. The WA one, I was quite impressed, had a lot of fungicides in it as well. So that was a really good resource and applicable across Australia. A lot of that. In Victoria, we've released our fungicide options here. I've put together a bit of a cheat sheet for people on field crop diseases that you can search for. Or you can look at the APVMA website, but start with your agronomist. And then if you are the agronomist, some of these other resources are there as well, or your peers. So talking about fungicide options, I don't want to go into this in too much detail, but we're losing a lot of fungicides in our cereals. But when we look at pulses, we've got a whole range of fungicides that are available to us. That gives us really good options to rotate between our fungicides, essentially. So I'll put them on there. I haven't listed the pathogen or disease that they basically cover, but it just shows you the crops and what fungicides are available, and you can look at that in more detail. Some of the watch-outs, though, with our fungicides, bixafen prothioconazole is basically, or aviator, basically the latest you can spray that fungicide is early flowering. And it's basically late flowering for lupin for sclerotinia, or first pod is the BBCH69. So a lot of people go, oh yeah, it's last flower. And a lot of growth stage charts say last flower, but essentially when you go to BBCH70, that is first pod. And so where the plant is up to, it's first pod. So that's BBCH70. So when you're seeing pods, basically that's off the table completely. For our lupins, but aviators off the table basically from early flowering, whereas your phytophlometrophin, you've got until the first pod. And then our azoxystrobin cyproconazole, which is Amistar Extra, is BBCH75, which is basically mid-podding. So you've got a little bit longer. So I'd recommend starting with those more expensive chemistries up front. And in a season like this, where a lot of areas have turned drier, you've got that early protection when it was maybe a little bit more rainfall events. And then later in the season, you're left with your other fungicides, such as chlorothalonil, carbendazim, prosimidone, which can be applied much closer to harvest. And a lot of those are 28 days from harvest, though. But you've got much longer in the season to be able to apply those products. And then when seasons turn dry, you just may not need to use those products. So just going back to our fungicide resistance management, it starts with our varietal selection. If you've got a good building block, you don't need to worry about as much of the other things. Then you're looking at your non-chemical options, stubble management, and crop rotation, hygiene, seed quality, and sowing time, and then relying last on our fungicides. So I'd just like to point out a few AFREN resources that are there that give a whole range of resources, fact sheets across all our crops. And then I'll pass over to Levy.
Thank you, Josh. Afternoon, everyone. I'm going to talk about a single disease this afternoon, and that is powdery mildew. That is quite important in mungbean production in Queensland and also New South Wales. So the results that I'm going to talk about come from a project funded by GRDC, and the results about fungicide resistance were funded through the Broadacre Cropping Initiative, which is a partnership between the Queensland Department of Agriculture and Fisheries and our university. Let me see if I can move the slide. Yes, I could. Mungbean powdery mildew and any kind of powdery mildew is such an obvious disease, it doesn't even need introduction. In case of mungbean, the exact same symptoms that you can see there on that photo made by Lisa Kelly from QDEF. So the exact same symptoms can be caused by two quite different powdery mildew species. One is called Podosphaera xanthii, and the other one is Erysife vigne, which was discovered here in southeast Queensland a few years ago. The yield losses can be up to 40% if the powdery mildew infection appears before flowering, if the environmental conditions are conducive afterwards, and if the disease is left unmanaged. And there are a few reports about the impact of powdery mildew on harvest management, particularly on desiccation efficacy. Amongst those many fungicides that Josh has just mentioned, we only have two to control mungbean powdery mildew, and this is the tebuconazole, the active ingredient and the product, so a DMI or a group 3 fungicide, and Veritas opti, which is a cocktail of tebuconazole and azoxystrobin. So we have a group 3 and a group 11 fungicide in Veritas opti. The current recommendation is to spray at first sign of the disease and then approximately 2 weeks later if needed. And what does it mean if needed? When is it needed? To decide this, there is an app, and I hope I can get to the next slide. Yes, there is an app that is available for free in Google Play and also in App Store. This app called Powdery Mildew MBM was developed by the Department of Primary Industries and Regional Development in Western Australia and funded by GRDC. It is a really useful, reliable, and believe me, it's easy to use app to determine if sprays against mungbean powdery mildew are economical at all. So the industry practice is sometimes to just apply a preventive spray against mungbean powdery mildew together with an insecticide, for example, and then this will be followed by 1 or 2 fungicide sprays just for sure. However, using this app, this decision support tool, people can actually decide if spraying is economical at all, depending on the expected yield, depending on the expected grain price that year, depending on the cost of production, spray efficiency, whether it is aerial spray or it's going to be done through a tractor and so on and so on. So I would strongly recommend to download this app and try a few scenarios even now when the mungbean production season is over, and just play a little bit with this app because I think you will like it. We have done a series of field trials in 2022, 23, and 24 this year in Southeast Queensland to validate and demonstrate the value of this app with support received from GRDC through that project that I have already mentioned. This is just, the next slide is just a table of our field trials organized in experimental small plots. And as you see, we had plots that were sprayed with tebuconazole only once. Other plots, tebuconazole applied twice. Other plots, even 3 times sprayed with tebuconazole. And we tried 2 rates of Veritas opti, the regular rate according to the label and the highest rate recommended by the label. And if I Yes, and let me just show you an aerial image of a validation trial. This image was put together by Neil Robinson from our center. Thanks, Neil. It's a wonderful image. I would call your attention to the controls, which are almost covered, painted in white because of powdery mildew. Those plots sprayed with tebuconazole only once. Visually, they don't really differ from the controls. And the greenest plots are actually those that were sprayed with tebuconazole 3 times and Veritas opti the higher rate, just visually. These experiments in 2023 were not ideal because the disease showed up after flowering, so it didn't really have an impact on the yield. However, when we rated the severity of the powdery mildew infections in different plots, it was quite clear that tebuconazole applied once didn't really differ from the control, and the best disease management, the best reduction in disease severity was achieved with Veritas High applied 2 times. I just want to stress that the app did not recommend any sprays in any of these circumstances because the disease showed up late, and basically no real yield protection was provided by the sprays. However, we did the experiments just to see if there's an impact on disease severity. And yes, there was, as you can see in this figure. So just to conclude, all the field trials that we have done from 2022, the conclusions were that those preventive sprays, usually with tebuconazole, were not economical at all. Basically, they are not needed. Also, the app recommendations have always resulted in the most economic disease management. Even though the disease looked quite severe at the end of the season, the yield was not reduced if the disease showed up after flowering at the green pod stage. And some of these trials were actually the first experiments that compared the efficacy of those 2 available fungicide products that are currently available against mungbean powdery mildew. So if we look at these plots, experimental plots, carefully sprayed according to the label, and you see they are quite ugly. They are covered in white, heavily infected with powdery mildew. Of course, the question is this a field failure of the chemical management? And I think everyone will agree that 100% of powdery mildew control in mungbean cannot be achieved with those 2 fungicide products that are currently available for this particular disease. So we looked at the DNA markers associated with group 3 fungicides, so DMI fungicides, and group 11 fungicides. And we did detect the DNA marker for DMI resistance in Podospheric Xanthia, in one of the causal agents of the disease, in just 2 paddocks since 2019. And we have detected the DNA markers for the QOI, or group 11 fungicide resistance, in both powdery mildew species that cause this disease, altogether in 4 paddocks. So this is not a lot. But certainly the DNA markers were detected. We don't have quantitative data about these DNA markers, but currently, as part of a PhD project, we are developing a qPCR, or a digital droplet PCR method, to provide quantitative data about the frequencies of these fungicide resistance cases. The fact that apparently the fungicide resistance markers are not widespread in mungbean powdery mildew populations means that we are still in time. We can still act, and we can still apply the AFREN recommendations to keep these 2 fungicide products useful and efficient as long as possible. I won't repeat these conclusions here, and I would like to just recommend a few further reading about mungbean powdery mildew in particular. There's an update, GRDC update paper written by Elisa Kelly and co-authors, and the paper in ground cover supplement about fungicide resistance in Queensland in general, and another paper about mungbean powdery mildew in particular in ground cover this year. To just look at the so-called fungicide resistance 5 recommendations in case of mungbean powdery mildew, let me just start with the The first recommendation, avoid susceptible crop varieties. Well, in mung beans, all currently available varieties are susceptible to powdery mildew to some extent. The good news is that the mung bean breeding team led by Mary Ryan at Hermitage Research Facility, they are working on new mung bean varieties that may have a higher resistance to this particular disease. Second recommendation, rotate crops, use time and distance to reduce disease carryover. Well, we are dealing with an airborne disease, so rotation won't help to manage mung bean powdery mildew very much. The third recommendation is to use non-chemical control methods to reduce disease pressure. Is there any non-chemical control method to manage mung bean powdery mildew, I ask? And yes, there is. Plant early in the summer season if you can, because at high temperatures, powdery mildew usually doesn't show up in the paddocks. And if the green pod stage is already achieved before the temperatures drop, late summer, early autumn, then basically the powdery mildew infections won't have an impact on the yield anymore. So this is the non-chemical control method in case of mung bean powdery mildew, just plant early. Fourth, spray only if necessary and apply strategically. That's why we have the app. So the app will guide strategic decisions about fungicide applications against this disease. And last but not least, rotate and mix mode of action group fungicides. As I mentioned, we have two mode of action groups, and both are efficient against mung bean powdery mildew, although the DNA markers of resistance are there, so these should be treated as red flags. Yes, the pathogen populations are able to respond to fungicide use. However, probably we are still in time to use these fungicide mode of action groups that we have strategically and preserve their efficacies. Last but not least, I would like to acknowledge all our industry partners who were part of the current GRDC funded project, and also those wonderful plant pathologists across the country who supported this project. So this is my last slide. Thanks for your attention.
Thank you very much, Levi, for that. I don't, there haven't been any questions come through, so we might wrap it up there. I just wanted to thank Fran, Levi, and Josh for presenting on today's webinar. If you do have any questions, contact details are all available on the AFREN website. I've just popped a link to a short survey in the chat box. If you could jump on and give us your feedback about today's webinar, that would be excellent. But yeah, on behalf of the AFREN team, thank you very much to Fran, Josh, and Levi for presenting today. Thank you to everyone for joining us, and we look forward to welcoming you back to another AFREN event in the very near future. Thanks, everyone. Oh, hang on, sorry. There is one question. Can off-target fungicide use be a problem in pulse fungicide resistance? Josh, is that one maybe for you? Sorry, the question is, can off-target fungicide use be a problem in pulse fungicide resistance?
Can it be a, hang on, I've just lost the screen, sorry. Let me just bring up the chat. So can off-target fungicide usage be a problem? Yes, definitely. It depends on what's being applied. Off-target, if you're within the label rate on the register, within the registered label rate on the registered crop, within the registered timing, and there's different pathogens there, then it's certainly allowed or permitted by the use standards of chemistry. But basically, if you're just continuing to apply a fungicide, I suppose we all went through that. If you don't need to be applying a fungicide, then yes, definitely it can lead to resistance over time. Yeah, I hope that answers it.
Yes, that is great.
It's very much a different answer whether you can permitted doing something or whether it's legally allowed to whether That's our recommendation following the AFREN principles, I suppose, is the easiest way to put it.
No worries. No, look, that's great. All right. Thank you, everyone. We might wrap it up there, but thanks again for joining us, and we will see you again soon. Thanks.